Welcome to the 54th InterScience Conference on Antimicrobial Agents and Chemotherapy, or ICAAC. I'm Michael Schmidt from This Week in Microbiology. It's my distinct pleasure to welcome our guest here this morning, Dr. Charles Gerba from Arizona State University, if I'm remembering right.
No, University of Arizona.
University of Arizona. I always get something wrong. And together with the help of the program committee, we have selected some of the science that's being presented here this week at ICAAC for a discussion up here on the couch. We think that the topics are provocative, and we welcome the scientists coming and chatting with us about their exciting results. I encourage you out there on the online chat room and in the Twitterverse, if you have a question for Dr. Gerba, please feel free to post it. And should you in the audience have questions, feel free to come up to the podium mic, state your name, and ask your question. Well, welcome, Dr. Gerba. Sorry I got your school all wrong. It was football week, and I should have known how the University of Arizona did. But I guess Arizona State was on the sports page ahead of it, so I apologize. You've done some really exciting things, and I'll give the audience a tease of what we're going to learn about. How quickly viruses can contaminate a building. This, of course, is a story that's blown up over the weekend. We've been watching the news about enterovirus Delta 68 that's blown up in the news. It's making big news because kids are getting sick. But, you know, I think the story that we're going to talk about this morning is right on point. And the story is how quickly viruses can contaminate a building. Using tracer viruses, your group found that contamination of just a single doorknob, so we're not talking rocket science, a doorknob or a tabletop results in the spread of viruses throughout office buildings, hotels, and healthcare facilities. Within 2 to 4 hours, that's just absolutely astounding that one single doorknob can move a virus through a building within 2 to 4 hours. And this can be detected on, and this was the frightening piece for me, between 40 and 60% of the workers. So you were able to recover virus from these folks or visitors to the facility. So it really gives a whole new meaning to it. So why don't you share with us how you got the idea to look at these things, and more importantly, do you have a solution?
Yeah, what we wanted to really do is actually develop kind of scenarios, not only to learn how viruses move in these kind of environments, but it allows us an opportunity to look at interventions. What happens if we provide people with hand sanitizers or disinfectants to use in these environments? Would that reduce the spread of the viruses and the probability of people being exposed to them? One of the big advantages of this type of work, we can quantitate it very well by putting a virus that doesn't infect people on certain objects and see how it moves around. You already pointed out how we actually put a virus on a push plate to an office building of 80 people, had 3 entrances, and in 4 hours it ended up on over half the people's hands. It ended up on over half the surfaces that people touch in that building was a thing. And what we really learned was really the hand is quicker than the sneeze and the spread of disease, to really tell you, based on what we saw in this. And you think about the dynamics of an office building, I think, and how people move around. What we learned was the first area to be contaminated was the coffee break room. Because what's the first thing you do when you go to work? You get a cup of coffee, right? Yeah, you want to be the first one in that coffee break room every day, from what we've learned. And that actually turned out to be a hot spot, was the coffee break room, because a lot of people go in there. More of a hot spot than the restroom, which a lot of people might blame in that.
So it was the meeting area. It's both the meeting area for us folks who can walk and talk.
That's right.
But it's also the meeting area for the viruses that are mingling, and probably bacteria that are going to exchange antibiotic resistance traits.
Yeah, that's true. And you have to, every time you touch these surfaces, you're picking up between 30 and 50% of the organisms that were on that surface.
Wow.
And a lot of common diseases like diarrhea, colds, flus are actually spread by your hands when you touch your face. Now, the average adult touches their face about 3 to 4 times every minute, once every 3 or 4 minutes. So it's a very frequent thing, and every time you do that, you touch your nose, mouth, or eyes, you have a probability of transmitting disease. So that's how the kind of the dynamics work of you picking up these organisms and then having a probability of getting infected with them.
So how did the people react when they learned that half the building was contaminated with the virus from the front door, side door or back door within 2 to 4 hours? Were they upset? Did they begin to use hand sanitizers more frequently? Did they yell at the cleaning staff? What went on?
Well, really, they were quite surprised because they said, well, I never meet the people in the other part of the building, so how could I possibly have come in contact with it? Well, you don't have to. And I think that's what we have to realize, how important the hand is in that. Because, like, for example, if you sneeze in your office or you break that sneeze, nobody else is exposed, but you get it on a surface, you get it on your hand, and you go to the bake room and you contaminate it.
So when they bless you when you get sneezed, it's actually controlling that spread.
That's right.
So the blessing actually works.
Right, yeah. Maybe it's a prayer, it won't move into your office, but the probability from what we've seen is very high. But what really another object was to look at what kind of interventions would reduce the risk of the movement of the viruses and the exposure of the people. So we used several different types of interventions. One, the alcohol gel sanitizers, quaternary ammonium disinfectants, or quats we call them, which we know were effective against viruses like noroviruses, the cold viruses. And we talked to the people in the office building, and then half of them agreed to use hand sanitizers or disinfectants in their office area. And then we came back and released the viruses again. And we found a dramatic decrease in the occurrence of the viruses, about a decrease of 60–80% of the surfaces were less contaminated, and a 99% decrease in the amount of viruses that we found on surfaces in these buildings. So we had a rather dramatic effect. What surprised us was that only half the people agreed to use hand sanitizers and disinfectants, yet we had a very dramatic effect on the spread of the viruses in the office building.
So how often do they have to, did you prescribe how often they had to use the disinfectant or the hand sanitizer? For example, every time you went into the break room to get coffee, you had to return to your desk and hand sanitize, or you had to hand sanitize before you went into the break room. Did you give them any instruction? Yeah,
we suggested that anytime they've been out in public that they use a hand sanitizer, they use a disinfectant wipe on their desk once a day, and we put a hand sanitizer in the break room too, so there could be a dispenser there so they could actually get it. So that's make it convenient is what we really did for it. And I'm sure a lot of people who weren't involved or said they wouldn't use a hand sanitizer or disinfectant also participated because it was there and made available to them.
So were people concerned about other frequently touched items in their office space, like their telephones, their keyboards, their mice? I know a lot of folks do have communal offices where they're out on the road and they may come back to a workplace and 4 other people use that same space. So were they concerned about that, or were these people who always used the same spots?
No, they were usually people who had their own offices in that. Okay. So they really didn't meet a lot except at meetings or the break room and that. It's kind of interesting, though, if you look at office buildings in the fall during the cold season, about 1/3 of the surfaces you'll actually touch have a cold virus on it, we found in other studies. So it's more frequent than you might think, because a lot of people come to work with a cold or flu, and then contaminate these areas and that. So
do you think since this enterovirus that's been blowing up over the weekend across the Midwest, do you think it's because school started?
Yeah, definitely. I think it's because school season is in. We've done studies on virus occurrence in schools, actually, and you'll find commonly cold flu viruses on desktops in schools and that. And we actually did interventions there, too, looking at quaternary ammonium disinfectant wipes for school desks once a day, going into schools during the cold and flu season. We found we could reduce absenteeism by about 50% if kids would just disinfect their desks once a day. So a real little strategy here of just using these on some regular basis seems to really reduce the risk of exposure and absenteeism, certainly in schools.
So as parents are concerned about their kids, just watching the morning news shows this morning, it was pretty frightening. We had kids in hospital beds, so mom and dad may just want to send their son or daughter to school with a little disinfectant wipe and tell them to run it over their desk surface and run it over their hands before they eat lunch.
Yeah, I think that's a good idea. Certainly, when we've done virus release studies, that makes a big difference in the occurrence and the exposure of children to viruses. And they bring their hands and face a lot, their fingers to their face a lot more. Oh, they're all the time. You know, small children do it as much as once every minute. If you actually look at small children.
And the toddlers who are crawling, there isn't something that they didn't want to put into their mouth.
Right. Yeah. Almost everything goes in their mouth. They figure out what food is.
Yes.
Yes. So you see that too. You know, I think relevant to this, we've been actually having the opportunity to study hotels too, where we put a virus in a hotel room, like on a nightstand or a desk or a doorknob, and see how far it spreads in a hotel, which always makes me worry now that I've done these studies, because what we found, within a day, the virus goes to the next several rooms that the maids clean, because when they're cleaning with towels and that, they'll pick up a virus and move it from one room to the next. And we actually found the virus actually moves to conferences from the hotel rooms of people attending a conference or not, within a matter of a day. So really, if somebody's sneezing in the room next to you, there's a good chance that virus, if that's what he has, will be in your room the next day.
So, since a lot of our attendees are going to go to ID Week in a few weeks, we'll bring the viruses from ICAAC to ID Week with us.
Yeah, if you're still infected, you certainly will in there. And then again, we did studies where we provided maids with disinfectants that would kill the viruses that we were studying, and it was very effective in reducing the risk to the people. So a lot of the moral of this comes out, you know, things that we recommend like hand sanitizers, hand washing, disinfecting wipes, disinfectants, do their job. They reduce our exposure in many cases by 90 to 90%, and our probability of getting actually infected by 80 or 90%. So by doing these kind of virus release studies and looking for where viruses occur, we can get a more precise idea of the benefits of these types of really weapons in germ warfare today.
So the virus that you used in your study, what was it?
We use a bacterial virus called MS2.
So it's not going to make people sick,
no.
It's just going to make bacteria sick.
That's right. It doesn't affect plants, animals, human beings, but it's been used as a model for like noroviruses because it's a very hardy virus, same size, shape, a lot of resistance, similar resistance to a lot of disinfectants. So that's why we chose that as a model virus.
So what's next?
Well, we actually want to do more detailed studies actually in public restrooms. One of our questions is if we contaminate a public restroom with this virus in a facility, where will it end up? Will it end up in an office building? How will it travel on it? If I put a virus on a floor, do I have to worry about it? We know that the bottom of women's purses pick up bacteria from restrooms floors. So we want to see, will that move a virus around an office, or could he be able to take it home? I think one of the things people seem to fear most is restrooms, but we don't have really a lot of quantitative data on how important that is and how much exposure you have in a public restroom. So we want to contaminate different objects and see if you really can pick up a virus that causes an illness in a restroom, where would it be and what type of interventions would work best to prevent that?
One of the interesting things is how few viruses it takes to make us sick. And so what concentrations are you typically looking at when you walk into a contaminated environment? Is it millions, is it billions, or is it simply something like 100?
Well, some studies we've done indicate it's more in the range of 100 to 1,000.
So not very many.
Not very many, no.
And since they're so small, it's really exercise in did you win the lottery in order to pick it up and touch it. But it's probably because we all touch the same part of things. Like
That's right.
If you look at push plates to doors, you can see the shiny part where everyone was touching it.
Yeah, really, we're in a generation where we touch more surfaces that more people have touched than any time in history, because we're spending 80% of our time indoors. So you don't have to reach out very far to touch anybody, because you're going to come across a lot of their viruses all the time. It only takes, for many of these viruses, 1 to 10 to cause an infection. So you don't have to really be exposed to many, but because of the way we live today and spend so much time indoors, we're actually in contact probably with more germs from other people than any generation in history. Go back 120 years when most of us were farmers, we went into town once a week. Might have been exposed to a few people's germs, but today, like at this meeting, we're going to be exposed to maybe thousands of people's germs that will be going to deposit on all the push plates and other surfaces we're going to be touching.
And people wear gloves then. too.
Well, we see we don't have to get that desperate. By the interventions we've been doing, just hand washing, hand sanitizers, disinfectants properly used in key places, can cut these risks dramatically.
You're watching ASM Live, and I'd like to invite questions from either the audience or those of you in the online chat room. We have a question from the audience, please.
Yeah, good morning. Nathan Seppa at Science News. A lot of work has been done looking at the transfer and how you can pick up viruses off doorknobs, and this part has been established. I take it what's different with yours is you controlled for this. You planted these in a specific building and then tracked this. And the other thing I'm just curious about is how many buildings did you use? Can you give us some of the actual data in the numbers of people?
Yeah, that's a good question. You're very right when you say the numbers are available on transfer of this virus too. We've studied, and you could model this, but what's very difficult is modeling human behavior. So that's why we added the virus to these key surfaces to get that whole picture of how these viruses move in a building rather than just doing a model, just as a real building. We used 2 offices. One office had about 20 people in it, and the other office had 80 people in it. And we ran these tests several times to make the data statistically significant.
Another question from the audience, please.
Yeah, sure. Michael Smith, MedPage Today. I've been to this meeting several times over the last many years, and almost every year somebody comes in and says, "Don't touch the light switch in your." You might even have been your colleagues or you have come in and said, "Don't touch the light switch. Don't touch this and that in the hotel room." What's new about this study that doesn't? We already knew that viruses and bacteria were all over the place and were being spread.
Yeah, what was different about our study in terms of looking at your exposure was we released an agent and we quantified it. What we did is we looked at the numbers, and we actually ran models to tell you what your probability of getting a disease would be in an office building. It was quite high. If you were a susceptible individual, you're going to be close to 80%. But then we did interventions. We saw we could reduce that probability by 90 to like 5–10%. So we were able to do a quantitative picture. The other thing you discover is that light switch isn't so important. Why? Flip it on once at the day and you flip it home on the way out the door, right? Not many people are touching a light switch. What they're touching is the coffee pot handle. What they're touching is the microwave, open the microwave up. What they're touching is the desktops, the areas where they eat all the time. Their phone is one of the most, phone and desktops and tables are the most heavily contaminated, by the way, is what we found out. I mean, most people are talking dirty and don't realize it just based on the number of organisms we found on a phone.
Talking dirty and don't realize it. That's an interesting quote.
Can I follow up on that? You said the probability of disease is 80%, and I'm a little puzzled by that. Do you mean the probability of contamination, of colonization with this virus?
That's right.
This is not a virus that causes disease.
Right. The probability of you actually getting infected by the virus.
Okay, actually carrying it along with you.
That's right. You can model the probability if you know the occurrence of the virus of it getting to your mouth or nose, and then the probability of it actually infecting you. So you can get an idea how many people in that office over time would get infected.
On the other hand, most people, most of the time, don't become ill, even with pathogenic viruses. Presumably, there are other factors coming into play.
Yes, there is, like pre-existing immunity. They've already had the virus. They're depending on their personal habits. You're really running a gamut all the time when you're going to an office building, and it's a gamble. The whole idea is to keep the odds in your favor and not the germ's favor. And that's what a lot of this is about, how to keep the odds in your favor during the cold and flu season.
Okay, and one final one, if I may. The sort of abstract of this that I read talked about quaternary ammonium wipes as opposed to hand sanitizers. But you mentioned both, and in fact, you seem to emphasize the hand sanitizer. So can you quantify that again? What's best? Are they equivalent?
They're pretty much equivalent in a lot of the quaternary ammonium quats are effective against norovirus and cold and flu viruses, and they were quite effective. We've done studies where we looked at just the wipes by themselves and a combination of both, and both of them do really well from what we've seen. And I always look at those are two of the greatest interventions that the general public can use, and what we really wanted to get quantitative data on how effective, and both were very effective.
Can you
And in
quantify that?
combination and by themselves.
How would you quantify that?
Oh, I would say your reduction in exposure with either one of those is about 90 to 99%, depending on your environment, whether it's an office building, a home, or a school.
Thank you.
So, Mr. Smith's question prompted one in my own brain. This is sort of an inside baseball type of question, but how did you measure whether or not I got a virus? Did you have me roll my hand across, soft auger or what?
Yeah, to determine whether you got a virus, we actually monitored people's hands too. So we knew the concentration of viruses on their hands. And so by knowing that, we could predict how often they bring their fingers to the nose, mouth, and eyes, about 16 times per hour, and what their dose was. So then we could calculate the probability of getting infected.
Okay. There was another question?
So I just wanted to clarify that the way you calculated the value of the interventions, of the wipes and so on, did this bacteriophage, this substitute virus, innocuous virus you use, is that a good proxy then for these other more actually infective things? And does it also correlate then with the value of quats or alcohol?
That's a good question. Really, how good a model is this? We picked MS2 because it's been used as a model for like noroviruses, and it has a similar shape, size, and survivability of like rhinoviruses, noroviruses. And we tested to make sure the hand sanitizers and the quats were effective against this virus before we did the studies. So we wanted to ensure that it was. And like with noroviruses, there's about 60 different quat formulations that are effective against noroviruses, so we knew it worked against that too.
And one of the things I forgot to ask you is, when is your poster or paper today?
Oh yeah, the poster on this, summarizing all this work, is at 11 o'clock today.
Down in the poster hall?
Down in the poster hall.
Okay, and you're probably going to publish this fairly shortly after this meeting, so those of you who aren't here at the meeting, you can have an opportunity to hear about what Dr. Gerber's been up to with this exciting trial. You've been watching ASM Live here at ICAAC, and I'd like to thank my audience for their participation in this edition. Please join us again in a few minutes when we continue ASM Live here at ICAAC, or listen for me, Michael Schmidt, along with my colleagues Mozelio Schechter from Small Things Considered, Michelle Swanson from the University of Michigan, and the host of This Week in Microbiology, Vince Racaniello from Columbia University, when we all get together to have another edition of TWIM, the podcast that discusses the unseen life on planet Earth. Thank you, everybody, and we'll see you again shortly. Did I get everything?
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan. So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right? Remember, this building is basically a second building inside the main building.